Okay, so is this first slide visible to everybody?
Yes, ma'am.
Okay, so now we are moving on to the next topic, which is energy, as you see, energy and nutrient recovery during waste treatment. Now, let me explain as to what is the need for recovery of nutrients and energy. Now, during any wastewater treatment, you know that domestic sewage and also water, contaminated water is all connected, and there is a very high BOD load, as well as the nitrogen and phosphorus, etc., which could be released. Now, the source of nitrogen, especially in the domestic wastes, is coming from the food matter, which is already discarded into the dustbin. The food matter could be containing proteins and other nitrogen containing material, as well as in the water. There is a lot of urea and other things which are going on. So in these wastes, which are actually being moved to the wastewater treatment plants, elements like carbon, nitrogen, and phosphorus are released. And these are all, now today we are beginning to understand that any carbon containing compound, nitrogen containing compound like ammonium or nitrate or phosphates, for example, these are very important nutrients, because if they were in nature in the form of nitrate or ammonium or phosphate, they would be taken up by the soil bacteria or they would be taken up by the plants. So the question is that when it goes into the wastewater, is it possible to recover energy from the waste organics? So if there is, for example, carbon containing organic matter, is it possible for us to recover energy? For example, methane. Okay, could we have microorganisms or biological systems which can convert all these organic carbon into energy like methane, for example? Also, are there bacteria which can, for example, denitrify nitrates or nitrites which are released into the wastewater? Can you recover nitrogen from it, or from phosphates or other kind of organic phosphates, etc.? Is it possible to release phosphorus from these? Otherwise, this material will be lost and nitrogen and phosphorus will not be returned back into the respective cycles. Okay, so that is the entire rationale behind either energy recovery or recovery of nitrogen and phosphorus from the wastes. So is this understood by everybody? Because that is what forms the basis of these lectures.
Yes, ma'am.
So let me go over first with you what are the different, you can say, steps which are involved in the conventional wastewater treatment plant. Okay, so here is a picture because it will give you an idea as to how the waste is processed, that is the domestic wastes, and then where exactly, when you say that whatever is left behind after the wastewater treatment is carried out, there is a lot of sludge which is left behind, and we will see as to where carbon, nitrogen, and phosphorus are present, so that the sludge can be reused for recovery of these nutrients. So the entire process of wastewater treatment can be divided into two sections. First is called as a primary treatment, as shown here. The second part is called as a secondary treatment. What are the steps in the primary treatment? So you see here the raw sewage is coming. The first thing that happens is that it is passed through certain screens. So screens, as you can see, for example, these could be like filters which are going to retain, for example, very large particulate matter, like you see the brown dot here is going to be retained, whereas the rest of the material will pass through the screens. Okay, so that is called as screen, for example. And after that, it comes into what is called as a comminutor. All right. So what you will see here, for example, are these two wheels which are rotating, and the purpose of this step is explained here. It is used to grind and shred the debris which passes through the screen in order to give it homogeneity. Okay, because eventually if the particulate or if this particulate matter is of a different size, the chances are that eventually during the secondary treatment, much of it is going to get settled at the bottom of the aeration tank. Okay, so you want to homogenize, you want to make sure everything is of a uniform particle density. And for that purpose, this is now passed through this, and it's explained that you grind it, etc., and eventually it is now collected into what is called as a grit chamber. what happens in this grid chamber? These are generally very long and narrow tanks. And what you see here is that a lot of this particulate matter, etc., is quite homogeneous, but still it is allowed to settle. The residence time in this section is approximately, 2 hours is for the primary clarifier, but it is passed over long tanks. Okay, during this process, all this particulate matter, for example, is going to settle down, and the rest of the liquid part is now going to move on into what is called as a primary clarifier. Here, the residence time is about 2 hours. Okay, so what happens at this stage is that a lot of this material, for example, you see at the bottom is going to be collected out, and this is called as a raw or a primary sludge. Whereas the clear part which you see on the top is now going to be passed, and from here you are moving into the secondary treatment. So it is passed through an aeration tank. All right, so have you looked at, I think if you have done some other wastewater treatment course, you must have seen already this part. So students,
Yes,
quick, you must have seen it. So I'm just kind of going to review it because, we are going to come back to it over and over again. So this is the aeration tank, and you have an air compressor through which air is passed in. And the idea is to only supply, there are specialized group of microorganisms which are present, about which I'm going to tell you. To them, you are going to provide oxygen, for example, air, of course, oxygen is what is important here, and the residence time in the aeration tank is of the order of 6 hours. Okay, so these bacteria are going to oxidize, for example, the organics, etc. That is, the BOD load is going to be reduced. All of these organic compounds are going to be oxidized into carbon dioxide and water through a lot of microbial activity, which you see here. Okay, and the residence time is of the order of 6 hours. And from here, it moves into what is called as a secondary clarifier. Again, any kind of a solid, and particularly the cells and some other solids are going to be left at the bottom of the clarifier, whereas the clear water, which is relatively more clearer than the input stream. And this is called as a secondary effluent. At that time, a disinfectant like chlorine, tablets, etc., can be added to it, and then it is discharged into the surface water. However, if it needs to go as drinking water or something, then of a further refinement, then a tertiary treatment is required, and that is how the secondary effluent is discharged. Okay, now this particular residue that you see here is called as activated sludge, and that is the reason why this treatment is also called as an activated sludge treatment process. So what you see here, for example, so this sludge comes here, and approximately 30% of the activated sludge is processed back, and it is returned back into the aerobic tank, into the aeration tank, because it is basically you are going to seed the aeration tank with already an activated microbial community. That is what the purpose is there. And the remaining, for example, sludge is going to be combined now with the raw or the primary sludge. So you have then sludge treatment and disposal. Okay, so is this overview clear to everybody?
yes, ma'am, it is clear.
Okay, so now in the activated sludge treatment, you are trying to treat the sewage or the industrial waste waters using aeration and a biological flock. Okay, now that means, when you say like, a microbial community or a biological flock, what all do we have in there? We have, for example, the protozoa. Amoeba is a very good example of that. Or you have other kind of microbial groups which are here, and they are all motile, etc. So this activated sludge, for example, contains both bacteria as well as these protozoa, which are able to bring about these oxidative activities. All right, so here is a picture of if you were to take what kind of a microbial community is there. There is a microscopic picture where, some kind of cells are just shown to you here. Okay, so that is how it operates. Now, the question is that when you talk about, when you talk about how much of sludge is accumulated, I will give you an idea of what is the extent of sludge which is generated. And this sludge is still rich. Even after the aeration and other treatments, there is still a lot of organic carbon left in there. there is still a lot of nitrogen and phosphorus which is left there. Okay, so the idea is that whether this sludge, because it is estimated that 50% of the total cost of running a wastewater treatment plant lies in the sludge treatment, because enormous amounts of sludge is generated because of this activity. So you have to spend a lot of money in order to treat this sludge or to dispose it off properly. You know, it's an ongoing activity. Domestic sewage or industrial wastewaters are constantly collected. So you have to constantly dispose of this sludge. Okay, it's a very, very expensive business, and you're also losing very important organics, nitrogen and phosphorus, etc. How much sludge is generated? Just to give you an idea, that the amount of sewage sludge which is now produced from the activated sludge process is obviously directly proportional to the amount of wastewater which is treated. So the more, the larger the volumes are, the more the sludge which is going to be generated. And in the second point, it says that the total sludge which is produced is, it contains both the primary sludge which you saw from the primary sedimentation tank, and it also contains the waste activated sludge from the aeration tank. So when you look at how much of waste activated sludge is produced, it is about 70 to 100 kilogram of waste activated sludge is produced per megaliter. Okay, so roughly about 80 kilogram on an average. If you look at the number, about 80 kilogram of sludge is produced per megaliter of wastewater which is being treated. Okay, now in addition to that, so here we are talking about the waste activated sludge, but the primary sludge is also accumulated, which is of the order of 110 to 170 kilogram of this primary sludge is also produced. Okay, so you know that you are dealing with large amount of sludge. Now, before we move into what should finally be done with the sludge in order to recover the nutrients or in order to recover energy, let us see what are the different kind of routes which are already available to us. So now the lecture is now going to discuss about recovery of energy, which is biogas or methane from the organics which are still left behind, and recovery of nitrogen and phosphorus. So when we look at the different sludge disposal routes, okay, so what is it? Obviously, the biological processes have come into picture only in the last, let's say, 10-15 years. But even before that, wastewater treatment activity is going on for a long time. So how was the sludge being disposed? Okay, so it could be used, for example, in agriculture because it contains nitrogen and phosphorus. So therefore, it could be applied as a fertilizer to the field. However, before you could do that, what are the kind of operations which are required? Because, you know, can you use sludge as it is, or does it need to undergo some kind of a processing before you can apply it in the field as a fertilizer? So the first thing that needs to be done is thickening. So T here stands for thickening, which is to approximately 5 to 6% of the dry weight solids. It is still, when it comes out from the aeration tank, you know, either the primary settling tank, it contains a lot of moisture, and in that form the sludge cannot be used, so some water needs to be removed. So the percent dry solid should be to the point of 5 to 6%. Okay, so this is weight by volume. So I hope that that part is clear to everybody as to what we mean by thickening. Is this clear? Is this clear to all of you?
Ma'am, I saw 5 to 6% should be the solids or water in that.
Yeah, it is like 5 to 6% gram per 100 ml of water. That is what we mean by dry solids. Okay, so 5 to 6 gram of solids per 100 ml of your material, of the liquid. So it's only in that form that it can be applied in the land as a fertilizer. Now R stands for road transport, because wherever the wastewater treatment plants are, from that place all the way up to agricultural land, wherever farming is going on, all this sludge needs to be transported. Okay, and remember that is also an expensive business, because you need to hire somebody, and the vehicles are going to run on petrol or on diesel, for example. So that is also a cost which you have to incur. Okay, so that is an energy intensive process, and that's why it is listed here. Now, in many other cases, for example, again, T stands for thickening, MD stands for mechanical dewatering. For some kind of application, 5 to 6% is not enough, but rather you must have 25 to 35% of dry solids in there per 100 ml of liquid. Okay, that is called as MD. So mechanical dewatering needs to be carried out of the sludge and then followed by road transport. In certain cases, again, if you want to apply it for agricultural purposes, you can do the thickening. Then anaerobic digestion is carried out for methane production. And this methane can serve, it is an energy source. You can have, for example, you can light bulbs in there, or you can supply this methane to heat the boiler or to increase the temperature, things like that. Okay, so by AD, what we mean is that anaerobic digestion of the sludge can also be carried out. Methane can be recovered from there, and it can be used in the farmlands for different kind of practices. Or like is shown in the fourth part, for some groups, for some kind of agricultural practices, you do have thickening carried out, anaerobic digestion, and MD stands for mechanical dewatering, and transport needs to be carried out. Or the sludge can be directly used for landfill, as is shown here. Okay, even for that, many of these operations need to be carried out. So in other words, in all of this, what you see is that these are all energy intensive processes. They all need to be carried out. Okay, landfill is, of course, you just put it simply, so much of sludge is generated, and now it can be just packed back into the land. Or it can be used as a solid fuel, where it can be burnt along with many other things. For example, if you are burning with coal or something else, the sludge can also be deposited there, and it can be used as a solid fuel. It can be burned. These are the kind of operations. Or simply as ash in thermal power plants also, it can be used. And these are the different things which go on. Of course, what you see here are additional processes like ID. ID stands, if you see here, it is indirect drying to 85 to 95. So all the water is practically removed if it is to be used for as a solid fuel or as ash. Or I is simply incineration or co-combustion with other solid fuels in power plants, cement kiln, etc. So the sludge which is therefore generated from the wastewater treatment plants also finds so many other uses. Okay, so sometimes people have to weigh. Now, is getting energy going to be more beneficial for me? Or should I just apply it as a manure in the field, or should I simply burn it? Okay, so you have to weigh out both the environmental aspects as well as other aspects to know what is the best use for the sludge which you are generating. So is this part clear?
Yes, ma'am.
So now, why is it that you still want to talk about anaerobic digestion? What advantages does it present? First of all, you can, this gives you an opportunity. If you carry out anaerobic digestion, it gives you a chance to transform organic matter into biogas, which is 60 to 70% of methane. So you can convert it into energy. And number two, it also reduces the amount of the final sludge solid for disposal. Okay, if you were not to carry it forward for anaerobic digestion, then as you saw that the volume of the sludge which is released is large. Okay, so if you subject it to anaerobic digestion, the solids are going to be lessened, and most of the pathogens which are present in the sludge are also going to be destroyed. They simply don't survive there because there are other microbial communities which are going to take over, and the other bacteria which are particularly hydrogen sulfide producing, and limiting the odor problems associated with the residual matter. Because if anaerobic digestion is not carried out, then the kind of foul smell that this emits is also very huge. Simply because, the organic matter is converted into hydrogen sulfide and things like that. So the odor is very bad. Okay, so because of these advantages, therefore, it is preferred now that the sludge should be now taken forward for anaerobic digestion. Now, in Europe, it is anticipated that the annual potential of biogas production in Europe is estimated in excess of 200 billion cubic meter. So this is how much of biogas can be produced if all of that is, and that's a huge source of energy, and it's a clean energy. And plus, a lot of things which are associated with disposal of the sludge are also taken care of. Okay, so because of all of these reasons, therefore, it's becoming a very important process. So now let's look at the anaerobic digestion process. What are the steps which have to be carried out in order to make this sludge available for methane production? Okay, so I just want to, I want to pause here for a minute. And other than Ratnesh, anybody else in the class who has any questions? There are a lot of people who are logged in. Ashu, can you tell me what all we have just done in the class so far? Amritanshu, Abhishek, just logging in and then disappearing. Gopika Arora, you're all grown up. You're all grown up, and to behave so irresponsibly, you are just simply logged into this, and then you are just moving around, going around all over the place, and not being in the lecture room. Uh, so any questions so far, Ratnesh? So is it clear as to what we have discussed so far?
Yes, ma'am, it is clear, but like I would like to know if it is being done like practically.
Absolutely, absolutely. When we go through finally, I will also give you the examples where anaerobic digestion is already integrated into the wastewater treatment plants. And please send a message to all your batchmates that they are missing out on very important topics, and this will not do, just logging in and disappearing. I mean, I'm
Ma'am,
ashamed.
maybe they
I'm
are, but
ashamed to see. I'm ashamed to see that grown-up students, graduating students, fourth-year students are behaving like this. Just send a message to everybody, Ratnesh.
okay, ma'am.
There is no conscious on the part of the student that you are enrolled in a degree program and you have to be attending the lectures. Even from your home, you can't do that. So if we look at the overall process or the flow chart, the first step that you see here, for example, is thickening of the sludge. Okay, and the purpose of thickening is to increase the solid content. Advantages are given here to reduce the free water, to bring down the cost of operations per unit volume. Okay, because initially the sludge which is now generated is going to be very huge volume wise. So when you carry out thickening of this, you are going to reduce the overall volume. Okay, so that is basically the purpose. And this can be carried out by three different processes which are listed here. The first is by gravity. That is, you simply allow the solid material to settle down as a consequence of gravitational effects, or by flotation or by belt filtration. What do we mean by flotation? Now, flotation is a process whereby in this, because it is a mixture of activated sludge and a primary sludge. Okay, so there are denitrifying bacteria which are in this particular group, and these denitrifying bacteria, they will convert nitrate into nitrites and then into nitric oxide, into N2O, and to nitrogen during this process. Okay, gases are formed. As a result of these gases being formed, along with the gases, a lot of this sludge is also carried upward on the surface of the tank. Okay, so all you have to do is to remove the upper part of the scum. So that is also like a concentration, it's concentration by way of flotation. Okay, so these gaseous products of anaerobic nitrate reduction will cause spontaneous flotation of the sludge, which is right on the surface, and then it can simply be taken off, and that is also a way of achieving concentration. And finally, belt filtration. Now, belt filtration is a process whereby the liquid sludge, as you will see very carefully, it can be passed over a conveyor belt which is connected to a vacuum. And as the sludge is passed over a conveyor belt, there are vacuum pumps which are attached at the bottom, which are going to remove all the water, all the aqueous material. So sludge at the end of the conveyor belt, the sludge will have thickened. Okay, and you are going to be reducing the overall volume. So that is something which is, yeah, go on.
Is it the same belt filtration where we attach a kind of blade to remove the filter cake also?
Well, if I look into, I did see some videos. Yeah, it could be. I did see some videos on belt filtration. Yeah, absolutely. You could have some kind of a blade, and then it is constantly the sludge is being removed in order to make room for the fresh incoming sludge. Surely, it must be coupled.
Yes, ma'am. This was it. It was like
Excellent.
introduced by TRS sir in one of the
Absolutely.
courses.
Absolutely. In the wastewater treatment plant, he must have told you. So it is to achieve, just the purpose is the same, that you are achieving basically thickening of this sludge.
Okay,
Absolutely.
ma'am.
Okay. So now after this step, you have what is called as a sludge pre-treatment. Why? Because in spite of thickening, although the aqueous volumes have, come low, but then the sludge, for example, the pH could not be very conducive for what is to be happening during the anaerobic digestion process. So maybe it needs to be more alkaline. You may need to adjust the pH, for example, which is now favorable for the anaerobic bacteria. Low temperature thermal pre-treatment may be required, or mechanical treatment may be required in order to make sludge kind of uniform, okay, so that the microbes will find it very easy to grow. So all of these are called as the pre-treatment processes of the thickened sludge. Okay, and then it moves into a digester, which is the anaerobic digester, and I will show you what are the different steps which are going to occur in the digester. And as a consequence of this, like you see here, for example, the biogas is formed. This biogas, that is methane. Methane is the major constituent of the biogas, but there are hydrogen sulfide, carbon dioxide also in there. But in that form, it can still be used as a source of energy, and then it can be moved into the storage vessel where it can be directly used, or it can be upgraded. That is, you can strip, for example, carbon dioxide or H2S can be stripped, and methane can be stored in a more purified form. Okay, and then the residual sludge is left behind, because when all these anaerobic activities are going on, methane is produced, which you have collected, but then there is also residual sludge. This can be dewatered, and further drying, it can be used for combustion or other kind of land applications. Now in the digester, of course, there is a sludge, there is gas, there is also a liquid component which is left behind. This liquid component can go back to the influent of the wastewater treatment plant, or it can be subjected to denitrification. Okay, by denitrification, what we mean is that some of the nitrates and other things which are left behind, okay, they are going to be reduced, and nitrogen is going to be released, and that can be returned back to the atmosphere. Okay, so this is the overall process. And now let me show you what are the events which are occurring within this anaerobic digester. So this is something which I had shown you earlier also in one of the papers which we were discussing, that so I hope this overall scheme is clear to everybody. It's such a fascinating topic. You are missing out on so much of learning when you are not listening. So was it clear, like what you saw in the last picture?
Yes, ma'am.
Okay, so now,
Yes, ma'am.
when you talk about the anaerobic digestion process, what are the kind of materials, the biologicals, which are present in the wastewaters? Now, these are complex polymers. They are proteins, carbohydrates, and lipids because they are collected from the domestic wastewaters. Okay, you throw, how much of food, etc., or the vegetable peels and what have you, many of these things are simply thrown in the dustbin. They are collected. What do they all contain? They contain proteins, they contain carbohydrates, they also contain lipids. Okay, or oil could come into the domestic water, into the waters that goes down the sink, and it could be very rich in lipids, for example. All right, so the first step in the anaerobic digestion process is called as hydrolysis. Okay, hydrolysis means that proteins are going to be hydrolyzed to amino acids, carbohydrates are going to be hydrolyzed into sugars, and lipids are going to be hydrolyzed into long chain fatty acids. All of these reactions are carried out by proteases, by carbohydrate degrading enzymes like amylases, or you have sucrase, you have invertase, you have whatever, which can break down the carbohydrate into monomers, and by lipids, which are, for example, your lipases. Okay, they are all converted now into monomeric forms. All right, so whatever is shown in the oval shape, these are the processes like is explained here in the legend. So whatever you see in the oval, this thing, it is a sub process in anaerobic digestion. So we have a sub process like hydrolysis. We have a sub process, the second process, which is called as acetogenesis. We have the third process, which is called as acetogenesis. And then we have methanogenesis, which is of two types. It is either homoacetogenesis, or it is going to be no, I think we should just look at methanogenesis right now as a process. And of course, homoacetogenesis also occurs. Okay, so all of these are the processes which are going to occur. For the sake of convenience, for the sake of an overall understanding, we don't talk about too much about homoacetogenesis. We only say that there are four steps. First is hydrolysis, second is acetogenesis, third is acetogenesis, and fourth is methanogenesis. Okay, these are the four processes. What kind of molecules are you going to deal with? Are, for example, these are the chemical forms. That is, what kind of molecules are you dealing with are shown in the form of a rectangle, and that is why here this is a rectangular picture. Proteins, carbohydrates, lipids, amino acids and sugars, long chain fatty acids. They can be broken down into hydrogen and carbon dioxide, or amino acids and sugars can be broken down into acetate, or sugars can be broken down into propionate and butyrate. And that is why all of these are shown in the rectangular form. So these are the molecules, these are the products. And finally, you see here methane and carbon dioxide. Okay, that's the way you need to interpret them. In addition to that, what you see in these little circles are the group of bacteria which are carrying out the different processes. Okay, so when you see one, that means these are all a group of fermentative bacteria. Okay, and these bacteria, they can only work under anaerobic condition, and that is the reason why this is called as anaerobic digestion. Because as we are going to see, if the process was aerobic, then what happens is that amino acids and sugars will be completely mineralized into carbon dioxide and water. Long chain fatty acids would again be completely oxidized to carbon dioxide and water if this was an oxidative process. But since it is an anaerobic process, there is only partial oxidation which occurs, and this results in the accumulation of hydrogen, carbon dioxide, or you can have acetate, or you can have propionate and butyrate, etc. It's only because it is occurring under anaerobic conditions. Okay, so is that point clear to everybody?
Yes,
Yes,
ma'am.
ma'am. One doubt, like after acidogenesis, we learned like volatile organic compounds are produced. So this propionate, butyrate are examples of volatile organic compounds.
Absolutely. Absolutely. I'll show you in the next slide how they are made, and they are all called as volatile fatty acids because, the small chain fatty acids are all volatile. Propionic acid, acetic acid, butyric acid, they are all volatile compounds. And that is why these fatty acids are called as volatile fatty acids. Okay, so I'll show you in the next slide as to what is the biochemistry, and very, very simple biochemistry. Anybody who knows, I think, glycolysis and TCA cycle, that's all, the basic pathway. If you know that, then you will be able to understand how these different things are also occurring. Very, very simple to understand. Okay, so let me finish this particular picture. So first are the fermentative bacteria. Then you see the second group of bacteria which are circled as 2, and these are called as acetate forming or acetogenic microorganisms. Okay, why do you call them acetate forming? Because you see that amino acids or sugars, for example, instead of getting converted into volatile fatty acids, as happens with the fermentative bacteria, the second group of microorganism will directly convert it into acetate. Okay, that is the reason why these are called as acetate forming microorganisms. And similarly, the long chain fatty acids, etc., can also be taken up, okay, by this group of bacteria, which are the acetate forming bacteria, in order to give rise to acetate. And like you see here, in addition to that, there are, for example, the third group of bacteria, which are called as the hydrogen forming microorganisms. Like here, they can start from volatile fatty acids and they convert it here. The fourth group, homoacetogenic bacteria, which can use acetate and completely convert it into hydrogen and carbon dioxide. Okay, and since they only use acetate, they are called as homoacetogenic. They cannot use any other carbon source. They can't use propionate, butyrate. They can't use long chain fatty acid. They can only use acetate and convert them into hydrogen and carbon dioxide. So this group of microbes is very, very special, and it is called as homoacetogenic microorganisms. Okay, and eventually you have the fifth pathway, which is acetoclastic methanogenic microorganisms, and these bacteria, they start with acetate and they convert acetate into methane. But this is not the only way methane can be produced. Methane can also be produced from carbon dioxide and hydrogen by reduction of carbon dioxide by hydrogenotrophic microorganism. So it's very clear to understand what is the difference between fifth and sixth group of microorganism. Okay, these microorganisms, they start out with hydrogen and carbon dioxide, so they are called as hydrogenotrophic, and these microorganisms, they must start with acetate in order to make methane. All right, so this is the overall process. And so I mean, if you understand this particular slide, it's like you cannot forget anaerobic digestion process. Okay, it explains almost all the activities which are going on. Two things to remember here, that in the anaerobic digestion process, hydrolysis is the rate limiting step. Okay, so that is to say that all these different microorganisms are already present here. Once the monomers are present, once the long chain fatty acids are present, they can convert, they can take it forward. But hydrolysis is the one which is a limiting step. Okay, and in order to increase the anaerobic digestion process, therefore, there are strategies which are being developed in order to, in order to increase the rate of the hydrolysis reaction, so that more and more of these monomers are available to the bacteria and overall yield of the process can be increased. Okay, so now we take a minute and we talk about the microbiology of the process. As you see in the anaerobic digestion, therefore, extremely complex microbiology, very delicate, and it involves different types of bacterial groups like you saw here. Okay, now remember, each one of these bacterial group, they have their own optimum working conditions. They only operate at a certain alkalinity, at a certain pH, at a certain temperature, at a certain reducing power, and there should be so much of reducing conditions, etc. So when you talk about anaerobic digestion, therefore you have to make sure that all these conditions are satisfied for all these different groups of microorganisms. These groups are sensitive to, and they are possibly inhibited by several process parameters such as pH if it fluctuates too much, alkalinity, concentration of the free ammonia which is generated, hydrogen, sodium, potassium, heavy metal, volatile fatty acids and others. And the objective of putting down all of these factors in there, okay, is to make you understand that eventually when optimization has to be carried out, then all of these factors need to be individually addressed. Okay, you need to regulate all of these parameters because the overall process of anaerobic digestion is going to be affected by this. Okay, and in the last point, to accelerate the digestion and enhance the production of biogas, various pre-treatments can be used which improve the rate limiting hydrolysis step. Okay, like I have shown here, for example, in the rate step in this process is the hydrolysis step. So can treatment etc. be carried out before it is subjected for methane production? Okay, now if we try to look at, for example, what is the percentage, what is going where, here is a picture to show you. So starting with the complex organics, which you start out with right here, okay, nearly 76% of the organics are taken forward to give rise to higher organic acids. The 4% is directly converted into hydrogen, and nearly 20% is converted into acetic acid. Okay, then out of higher organics, approximately 24% goes into production of hydrogen and carbon dioxide, and 52% goes into acetic acid, and the rest is something which you cannot account for either in terms of carbon dioxide or something. Okay, and finally, through this route, 28% of the final methane is produced through this particular route, that is hydrogen plus carbon dioxide condensing together by this group of microorganisms, which are called as the hydrogenotrophic microorganism, and 72% of the final methane that you see comes from acetic acid side. That is, it is through these acetylactic methanogenic microorganisms. Okay, so this gives you an overall percentage of how the carbon is going to be flowing through this entire chart. Okay, so is this slide clear to everybody?
Yes, ma'am, it is clear. Also, like this describes like about a specific this paper, or like in this which we are what we are seeing is like 100% of organics are being converted into methane. So like that is also like practically feasible, or
Plus point. And of course, you know, there is still, you can't say that the sludge is not going to be generated, but the sludge volume is substantially reduced. And I don't remember the exact number, but they say almost 50 to 60% reduction in sludge occurs. And that itself can bring down the cost of final operation, because the sludge treatment is a very important part of the overall wastewater treatment. So if the volume of the sludge is substantially reduced, the overall cost of the process also goes low. Okay, now very quickly, because I think this is something very interesting, that is how many of these, carbohydrates, etc., can be converted into, they can be converted into sugars, but how various sugars, for example, can be converted by fermentative bacteria into propionate and butyrate, etc., is shown in the next slide. Okay, so what we are looking at here is the biochemistry during acidogenesis, that is when volatile fatty acids are produced. Okay, and many of these, some of them are obligately anaerobic, like Clostridium, for example, is one major genus, but Clostridium is an obligate anaerobe. Okay, but apart from that, there are bacteria which are facultative anaerobes. Okay, that is, they can survive in the presence of oxygen, but Clostridium cannot. So glucose, for example, you will see that right up to pyruvate is the normal glycolytic pathway. Okay, now in many of these bacteria which are a part of the fermentative bacteria, they have this enzyme 5, which is lactate dehydrogenase. Okay, so the only additional enzyme that they have is a lactate dehydrogenase, which can convert pyruvate into lactic acid. So lactic acid is a short chain fatty acid. Okay, similarly, for example, this pyruvate can be converted into acetyl coenzyme A through reaction, because in the aerobic pathway, this pyruvate is converted into acetyl coenzyme A through pyruvate dehydrogenase. But here we have a ferredoxin oxidoreductase. It's a very different type of an enzyme which can convert pyruvate into definitely acetyl coenzyme A, but at the same time in the process hydrogen is going to be released and carbon dioxide is also released. Okay, so you can see now that this fermentative bacteria is capable of making lactic acid, carbon dioxide and hydrogen. Okay, or some fermentative bacteria, for example, it can go up to acetyl coenzyme A, but then it doesn't get into the TCA cycle. All right, but what happens is that now it goes into this acetokinase, so you get acetyl phosphate, and then it is hydrolyzed to give rise to ATP and acetic acid is formed. Okay, so you see how lactic acid and how acetic acid, which are the short chain fatty acids, how they can be formed. So these are the same kind of, pyruvate you already know, acetyl coenzyme A you know. Now acetyl coenzyme A, for example, can also be, it can go through reduction to acetaldehyde and ethanol, finally oxidation to ethanol. Or acetyl coenzyme A can undergo series of reduction oxidation steps to give rise to butyryl coenzyme A, and you can get either butanol or you can get butyrate, which is a short chain fatty acids. Okay, so I think what really emerges out of this is that since the sugars are not going to be metabolized aerobically, in which case glucose would have completely ended up as carbon dioxide and water. But because fermentation is occurring, okay, they don't use oxygen, and you make products like pyruvate, you do make products like acetyl coenzyme A, but there is an internal transfer of NAD, ATP formation, okay, and these are all fermentation end products. Acetone, isopropanol, butyrate, acetate, etc. Nowhere do you see carbon dioxide coming. Okay, all these are partially oxidized products of glucose. Butanol, ethanol, and all of these are bacteria which are fermentative in nature. So is this clear to everybody? So please remember, it's like a key of this whole. When you say acidogenesis, you need to understand how lactate, how acetate, how butyrate are being generated in the process. Okay, and interestingly, many people also look at genus like Clostridium for getting butanol. Like, when you talk about ethanol as an energy source for burning as a petrol, a lot of people are also considering making butanol, because that is also a clean fuel. And butanol can also be made through microbial activities, like you see here. These are organisms which have these additional enzymes which can also make a butanol. So fermentations can be carried out, and butanol can also be retrieved from the supernatant, just the way ethanol can be obtained from Saccharomyces fermentation. Same way butanol can also be obtained through bacterial fermentation. And people are trying to improvise on that so that, these are all clean energy sources. So is this point clear to everybody?
Yes, ma'am.
Okay. Now the other importance is that the facultative, you see, when you say facultative, by facultative what we mean is that they are not going to be killed by the presence of oxygen. Okay, something like Clostridium is an obligate anaerobic organism, but there are organisms which are the facultative anaerobes, which can still tolerate traces of oxygen. And the facultative members of this group, they also help to protect the oxygen sensitive methanogens by consuming the traces of oxygen which can enter in the feed. Please remember this point. It's very, very important. Because when wastes and other things are directed into the anaerobic digestion plant, traces of oxygen can also enter. But because there are these facultative bacteria, they simply take up these traces of oxygen, so that now the methanogenic bacteria, which are oxygen sensitive, they don't have to face oxygen at all. They are constantly being removed by these other group of bacteria. Okay. So the last slide was the chemistry or the biochemistry of acid formation. And here we are looking at the biochemistry of acetate formation and methane formation. Genic means production. So production of acetic acid and production of methane. So if you are dealing with volatile fatty acids like propionate and butyrate, then for example, if you start out with butyrate, okay, butyrate can be directly converted by enzyme, which is shown by the fourth group of microorganisms, and the summary is shown here. Look into the table, and what you see here, that groups of bacteria, you don't have to remember them. You know, I don't expect you to remember the groups of microorganism, but what you need to look at are like Syntrophomonas is a group of microorganism which can convert butyrate into acetate. Okay, plus it can also make hydrogen, and that's the reason why we say here, for example, butyrate can give rise to acetate and butyrate can also give rise to hydrogen here by this group of bacteria. Similarly, if you are starting out with propionate, which is actually in the last stage, in the last slide you saw how propionic acid can be formed. It's a volatile fatty acid. So from propionate to acetate, you use the group of microorganisms, which are number 5. The reaction is shown here, that the propionate can be converted into acetate plus hydrogen plus bicarbonate, which is the same as carbon dioxide. So these bacteria are called as Syntrophobacter. So these are the reactions which are going to occur. Or acetate by homoacetogenic bacteria, acetate can be converted into hydrogen and carbon dioxide by the 6th reaction, and you have the chemistry of that reaction right here. If you have lactate, it can be converted as shown by the first group of microorganism. Lactate can be directly converted into acetate. Okay, so is this part clear to everybody? I think it's a very, very clear, very self-explanatory slide where you will see as to how short chain fatty acids can be converted into acetate, or they can also be converted into hydrogen and carbon dioxide. Okay, and finally, we have hydrogen and carbon dioxide, which can be converted into methane, or you can have other reactions which are occurring, like acetate can be directly converted into methane by the group of bacteria which is shown by 7th step here. So you see here, acetate plus hydrogen will be converted into methane plus carbon dioxide. These are called as acetotrophic methanogen. Okay, or you can have, for example, reaction number 8 and 9. So 8 and 9, you can convert hydrogen and carbon dioxide directly or format directly into methane. And those chemistries are shown here in 8 and 9. These are the hydrogenotrophic methanogens. Okay, so I hope that clarity is there for you now as to how, and please remember this, because I would expect that you will pay attention to this slide and you will see as to how. So is this clear to everybody, all these reactions?
Yes, ma'am, it is clear.
Okay, now there's just one other point which I want to emphasize in this slide. Okay, and that is the term syntrophy. Okay, now this is a term which is appearing under, so syntrophy is a reaction, is a reaction which finally occurs because there is synergism in thermodynamics between two groups of microorganisms. To give you an example, if we look at the reaction 4, go back to this reaction. So when you look at the conversion of butyrate into hydrogen and hydrogen, etc., by Syntrophomonas, this particular reaction has a delta G value of 48.3 kilojoules. Okay, so thermodynamically, this reaction should not occur. However, it reacts directly in combination with hydrogenotrophic methanogen. So what do hydrogenotrophic methanogen do? They will immediately utilize either the acetate which is produced here, okay, and hydrogen which is produced here. So with the hydrogenotrophic bacteria, so So the format or any other kind of a proton, for example, is released here. These protons are going to be picked up by this group of bacteria and they will carry it forward to make methane. So in other words, the products of this reaction, such as hydrogen, protons, are going to be constantly taken up by a second group of microorganisms. So as a result of which, the thermodynamics of the reaction gets pushed in the forward direction and the overall delta G of the reaction becomes minus 17.3 kilojoules. However, this is only with hydrogenotrophic methanogen because they are capable of combining and they are capable of using these products immediately. This phenomenon is called as syntrophy. Okay, similarly in the second case, when you have propionate getting converted into acetate, hydrogen bicarbonate by this bacterium, the delta G of the reaction is 76 kilojoule per reaction. However, if it occurs constantly, again with hydrogenotrophic methanogens, okay, then the products of this reaction, like all the protons which you have, okay, all the hydrogen which is being generated, they are constantly being removed in the next reaction. So the overall delta G of the reaction becomes minus 17.3 kilojoules. Okay, so such a phenomena in which, like you say, syntrophy here. So during methane formation, there are two chemical reactions which are simultaneously going on in synergism with each other so that the overall delta G becomes equal to negative. It becomes a favorable reaction. So such kind of syntrophy is commonly seen during methane formation, and that is what drives this reaction forward. So please try to understand all of that, what is so important about it and why is it so important during methanogenesis. So I have just tried to explain. Yeah.
In the reaction 5 and 6, the microorganisms which we have, their name is like Syntropomonas. So are these microorganisms only live in what is called in symbiosis with these hydrogenotrophic methanogens, or are these found even without symbiosis? And if they will be uh, without symbiosis, then how can they survive when they require 48 kilojoule per reaction, something like that, energy to produce things?
Most of these bacteria, okay, they are facultative anaerobic bacteria. That is, they can survive in the presence of oxygen and they can also survive in the absence of oxygen. When they are being supplied in the presence of oxygen, then butyrate can be completely oxidized into carbon dioxide and water. And that is how they can use butyrate as a source of carbon. Okay, because the objective is to be used to use a carbon source and to derive ATP and reducing power. These are the two things which you require in biological respiration: ATP and a reducing power. ATP is required because any biosynthesis which is occurring in the cell requires input of ATP. You also require reductive power like NADH, etc., because then when you build up proteins, for example, or when you build up carbohydrates in the cell, starting with simpler ones, you need to reduce the compounds, so you require NADH. Okay, two things can be carried out even under anaerobic condition. However, when they are completely in the absence of oxygen, then this is the only way that the butyrate can be used up as a carbon source, but still it doesn't finish their requirement for generating ATP and NADH. Okay, that's the reason why only during anaerobic condition they need to come together with a syntrophic microorganism so that this can be completely carried forward and they are able to get enough ATP and energy out of that reaction. They are not obligate anaerobes, they are facultative anaerobes. So two things, obligate anaerobic or facultatively anaerobic. So in terms of biochemistry, those two concepts need to be understood. Respiration needs to be understood in terms of either aerobic respiration. By aerobic, what we mean in the presence of oxygen or oxic condition. Respiration can also be carried out anaerobically when you can use nitrate when you can use sulfate, when you can use carbon dioxide as a terminal electron acceptor.
Yes, ma'am.
Okay, so in the next slide, again, the same thing is explained to you. And you see, that is the reason why when we talk about the first leg of the reaction, they are all facultatively anaerobic proteobacteria. Okay, that means they are not obligately anaerobic. However, given the condition when the oxygen is less and they can carry out glycolysis, they can carry out pyruvate, but since the conditions are anaerobic, they are going to respire pyruvate into whatever acids and other things which are generated, and they also generate hydrogen and carbon dioxide during this anaerobic respiration. Okay, ATP is produced. This hydrogen and carbon dioxide is immediately taken up by these methanogenic archaea. Okay, that is why they are called as hydrogenotrophic methanogens, because they use hydrogen carbon dioxide, which is released here completely, and they are going to produce methane, etc., etc. So when there is a coupling of these two reactions, which is occurring between two groups of microorganisms in order to drive forward a reaction, all right, that is called as syntrophy. So at trophy has every, the word is derived from the trophic level, the energy level, and syn means synergy. So when there is a synergism which is involving like two different when it comes to energy generation, ATP generation, okay, if there is a synergism between two groups of microorganisms, that is called as syntrophy. So between facultatively anaerobic bacterium and between an archaea, there is a synergism which is occurring. That's why we call it as syntrophy. And this is very common during methane formation. Okay, so please, it's a very important concept, and two diverse groups of these are all bacteria, whereas this is an archaea. It's like bacteria are completely different, and archaea are totally different. So I hope everybody understands, it's a very, very important point of syntrophy. Okay, so do you want to take a break for just 5 minutes? We will come back at 10:40.
Okay, ma'am.
Or should we continue and finish it off?
Ma'am, we can continue. Like it is already 10:35.
चलो ठीक है, we'll do up to 10:50 and then I'm going to so now if we look at, for example, now you have understood in anaerobic digestion what is the kind of a chemistry, what is the kind of microorganisms, and how syntrophy acts together in order to convert an organic into methane form, because the end product here is methane. You are getting energy out of the waste material, so the focus is on getting methane eventually. What are the factors which are important, which are going to affect the anaerobic digestion? Number one, the pH of the process is extremely important. That's the same as alkalinity and the ratio of volatile fatty acids to alkalinity ratio. Okay, now the reason why all of these are important is because if we look at the fermentative bacteria, they operate within a pH of 4 to 8.5, so it has to be slightly acidic, whereas the methanogenic bacteria, they operate best in a narrow range of 6.5 to 7.2. So anytime the pH is going to go outside of this range, it's not favorable for the methanogenic bacteria. So pH has to be controlled. Number two, the production of the volatile fatty acids, it also reduces the pH. Okay, during the early legs, when you have glucose which is getting converted into lactic acid, acetic acid, propionic acid, short chain fatty acids, that also reduces the pH, which could be very bad for the methanogenic bacteria because they operate around neutral pH. Then the reduction in pH can be counteracted by the activity of the methanogenic bacteria, which also produce alkalinity in the form of carbon dioxide, ammonia, and bicarbonate. So in other words, a balance between the fermentative bacteria and between the archaea needs to be maintained in the anaerobic digester. Okay, the system pH is controlled by carbon dioxide concentration in the gas, carbonate in the liquid phase. Okay, so once the process keeps going, you have to be very carefully monitoring it also to see as to, what is happening at what stage, so that appropriate controls can be taken at the right time. Acidification of the reactor can be carried out, or alkalinity can be adjusted, so that, all the groups of microorganisms are capable of multiplying. So all of these factors are important. The second factor which controls anaerobic digestion is obviously the temperature, because temperature affects the growth rate and metabolism and the population dynamics. Now, acetotrophic methanogens are very sensitive to increasing temperatures. The degradation of propionate and butyrate is also sensitive to temperature about 70 degrees. Okay, then temperature also affects the partial pressure of hydrogen in the digesters, hence it influences the kinetics of the syntrophic metabolism. Okay, because hydrogen, carbon dioxide are constantly removed in the syntrophy process, and since temperature is going to affect the partial pressure of hydrogen, it will affect the overall process. High temperature also increases solubility, enhanced biological and chemical reaction rates, increasing death rate of the pathogens, etc. And finally, the retention time in the bioreactor. Okay, what should be the feed rates? How long should the residence time be in the anaerobic digester? All of these factors, I have just given you a summary, but people have worked extensively over the years on this to see how these parameters are going to affect. Now, very quickly, so if these wastewater treatment plants which are existing, they need to be coupled with an anaerobic digester plant. What kind of digesters are currently in use is shown over the next two slides. Okay, either it can be what is called as a standard rate digester, where you can see a clear picture of this is the digester, which is now here could be the wastewater treatment plant. The thickened sludge is taken and it is now moved forward to a standard rate digester. Okay, and the residence time here is anywhere from 30 to 60 days. The sludge content is usually neither heated nor mixed. So sludge is simply passed in here. There is no further mixing which is done here. The biogas which is generated as a consequence of microbial activity, it does provide some mixing, but yet there is compartmentation into different layers. Okay, so over a period of 30 days, there is a compartmentation where the digested sludge being heavier will move down. Actively digesting sludge will be somewhere here. The supernatant settles here, the scum layer, and the gases are at the top. Okay, so this is a gas outlet where 60% is methane, the rest is carbon dioxide, hydrogen, and H2S. So at this stage, the supernatant is taken and it is returned to the secondary treatment. The digested sludge can be removed and it can be dewatered. It can be used as a fertilizer. Okay, so this is one kind of a configuration of a standard rate digester, or the second one which is taken is called as a high rate digester. Okay, the only difference here is that the sludge is heated and it is completely mixed. So what you see here, that there is a heat exchanger which is coupled, and there is active mechanical mixing of the sludge which occurs here. Now, why it is not done in the previous one, okay, is because all of these are energy intensive processes. So if you were to do stirring, if you were to attain the temperature, because obviously in winters, then the temperature would be very low. Then the efficiency of the anaerobic digestion process will also go very low. Okay, so in the second one, which is called as a high rate digester, the sludge is heated, completely mixed, the raw sludge is thickened, and the feeding is very uniform. A uniform environment is provided, as a result of which, okay, then the tank volume is reduced, and the digester will go on. The sludge is taken forward, whatever is left behind, methane is collected, and the gas outlet appears somewhere here. There is a third type which is called as a two-stage digester. Okay, in the first stage, it is exactly what you saw as a high rate digester. That is, you do have temperature which is maintained, then there is mechanical stirring and heating which is going on. However, part of it is collected in a second storage tank. Okay, because thickened sludge is constantly passed into the reactor. Then after a 30 days hold up time, okay, then part of it is now taken up and it is stored in a second vessel. There is no mixing or heating here. The process generates its own heat. Okay, or there is a two-stage digester which have been used. So now let me show you, for example, a real case study where we have an integrated wastewater treatment plant and biogas production. And this is in, it's not in our country. I'm sure, I didn't get much data here, but this is from US. So in California, out of 242 sewage treatment plants, already 10 of these are generating 38 megawatt from biogas, and another 12 they burn the biogas which is produced in the anaerobic digester to produce hot water to heat digesters. Okay, what about the 220 others? They are still, they are, this is also old data. Many of them must have switched. So now let me show you, for example, how an anaerobic digester can be integrated within the existing wastewater treatment plant. Okay, so this is something which I showed you earlier. This is the aeration tank, secondary clarifier. The sludge is here, recycled sludge. So this waste sludge is now taken, all right, and it is fed. So whatever you have here from the primary clarifier and from the aeration tank is taken and it is moved forward into a digester. This is the anaerobic digester where, for example, methane will be produced, and what you see here, the biogas is coming out and it can be used to heat the boiler, and high pressure steam can be generated which can be used for moving the microturbine and producing electricity. Okay, or the heat can simply go back and it can heat the digester, keep the temperature appropriate at 37 degrees or 42 degrees, whatever you think is optimum, and whatever is remaining at the bottom, okay, can be used as a compost now. Okay, so is this clear to everybody as to how an anaerobic digester can be coupled to the existing wastewater treatment plant? Is this clear to
Yes,
everybody,
ma'am.
this picture? Okay.
Yes, ma'am.
And finally, the sludge, what you get, from this digester, right at the bottom, the sludge looks as shown below, and it can be used as a fertilizer as it is rich in nitrogen. Because, many of these bacteria which are present, they do remove only a little part of the nitrogen in the form of nitrogen gas. The remaining nitrogen is still present in this, and that is why it can still serve as a source of fertilizer. And similarly, there is a second, this is an actual facility, again in Minnesota, USA, and the name is given here, and approximately 12 million gallons of sludge is generated per day of sewage. Okay, so many gallons of sewage is generated per day, and 4.5 million gallons of sludge is formed per day in this facility, and 75,000 cubic feet of biogas is produced per day in this particular facility. So if we look at, for example, here what is happening, so you have your usual, you have your the aerobic activated sludge process, and what we are beginning is the sludge is going to enter into this wastewater treatment plant digester. Okay, so this is not the aerobic activated sludge, but this is a digester which is used for anaerobic digestion. Okay, so methane is produced along with 1000 ppm of hydrogen sulfide. Excess of gas can be flared out if you can't consume it in the same day. So a lot of biogas can be flared and allowed to go into waste. But the remaining methane is now compressed, and then sour gas stripping. Hydrogen sulfide is a contaminant which you don't want because it doesn't make it a clean fuel. So hydrogen sulfide can be stripped out of this. Okay, and it can be used under pressure to drive micro turbines, and then you have 108 kilowatt production of energy can be used, which are driven by these micro turbines, or you have so much of heat which is generated which goes through a heat exchanger. Okay, and it can be used to heat the boiler, so much of heat, the warm water which can then be brought back to heat this, and or a cold water, there is a heat exchanger here. The cold water can be, through this heat, for example, it can be converted and it can be used for this particular purpose. Okay, so what you see here, therefore, that such kind of anaerobic digesters are already integrated into the existing wastewater treatment plants. And what you are getting out of it is energy, that is methane. So is this clear to everybody?
Yes, ma'am.
Yes, ma'am.
Okay, so quickly summary of things which we have learned in the lecture, that due to high level of organic carbon in the waste activated sludge, it can be used to generate energy as well as solvents. Why do I say solvents? Can anybody answer that question? So please remember that chemistry which I showed you. You know, the fermentative bacteria, they also produce acetone and butanol in those biochemistry. And if we can harvest acetone and butanol, these are very, very precious solvents. So again, the process is the same. That is, during anaerobic digestion, energy methane can be produced and you can have acetone and butanol, which are formed. The case of methane generation is discussed. It comprised of 4 stages, which are called as hydrolysis, acidogenesis, that is production of volatile fatty acids, acetogenesis, which is production of acetic acid, and finally methanogenesis. These are the 4 stages which are of anaerobic digestion. Factors affecting these processes were discussed. Overall, the process will convert about 40 to 60% of the organic solids can be converted to methane and carbon dioxide. The chemical composition of this biogas which is produced is 60 to 65% methane. There is carbon dioxide, hydrogen, nitrogen, H2S, and H2O is produced. Of these, methane is the most valuable because it's a hydrocarbon fuel and it gives 36.5 megajoules per cubic meter in combustion. The residual organic matter is pretty stable, but it is odorless and it also contains significantly reduced levels of pathogens. Reactor configurations which are coupled to standard wastewater treatment plants were also described. So this is what you have learned so far in the lecture. Now, the second point, I'm just going to start it here and let you know, we'll just talk for another 5 minutes that, so when you talk about nutrient recovery, so far we only looked at what is to be done with the carbon or the organic material, the food material, the proteins or whatever. Okay. But at the same time, we have not talked about how to recover nitrogen and phosphorus. They also need to be recycled so that these are not lost in the environment and they cause eutrophication. Because if the secondary effluent, which is released into the water bodies, the BOD could be very low, but if they still contain nitrogen and phosphorus, they can cause eutrophication. Okay, excess of nitrogen, excess of phosphorus, if it is released into the water bodies, it leads to algal bloom. And I have already explained as to what are the harms which are associated with algal blooms. Now, in addition to that, the nitrate which is released in this secondary effluent is a risk to human health, especially as a possible cause of infant methemoglobinemia. That is, high levels of nitrate or nitrite can be toxic to newborns, causing anoxia or internal suffocation. Commonly, a bluish color to the skin is imparted, particularly around the baby's eyes and mouth. These symptoms of nitrate toxicity are commonly referred to as the blue baby syndrome. Okay, so even if the waste waters, etc., are treated, but if they contain very high level of nitrates or nitrate, okay, it is still very poisonous. So a pre or a post-treatment process, that is with respect to the activated sludge treatment, is therefore required in order to remove nitrogen and phosphorus from the effluent. Okay, and that's the reason why it is necessary. So not only is it toxic, but it also leads to eutrophication of water bodies. You can have algal bloom, you can have unnecessary algae and other kind of bacteria which are growing in water, which decrease the oxygen availability, etc., to other trophic levels which are in the aquatic water systems. So just to give you, the concern for high levels of nitrogen in the effluent has been recognized. So what is the source of this nitrogen? High nitrates which are in the soil is largely due to, because you see, although we are talking about soil here, but remember that because of rainfall, because of irrigation and other practices, even if there is high nitrate in the soil, eventually it can end up in the water bodies and it can cause pollution. Okay, so the high nitrates in the soil are because of natural biological processes, and they can also come from rainfall, from animal manure and nitrogen fertilizers. The US Environmental Protection Agency, UPA, which sets the standard, has established that in the drinking water, the level should not be more than 10 milligram of nitrogen per liter. If the level exceeds, then alternate waste source is required. And I have already discussed what are the problems with the excess of nitrate in the previous slide. Reactive nitrogen such as nitrate or ammonium, which is present in the surface waters and soil, can also enter the atmosphere as the smog component of nitric oxide and the greenhouse gas N2O. Eventually, this atmospheric nitrogen can be blown into nitrogen sensitive terrestrial environments, causing long-term changes. Okay, so therefore, there is the fear of pollution, there is the fear of eutrophication, so on and so forth. And that is why increasingly today, people, we are becoming aware, or the guiding authorities, the environmental agencies are becoming aware, and they are setting up new standards for release of nitrogen into the environment. Now, what kind of a treatment needs to be given will depend on the initial concentration of the nitrate. Okay, and Mulder, who has done a lot of work in this area, is an authority, etc. There are 3 concentration ranges which have been identified. So if the level of nitrogen is less than 100 milligram per liter, as is usually the case in the domestic wastewater, then a biological nitrogen removal procedure needs to be targeted. Okay, so is this clear? So you are not advised to use any kind of a chemical process if the level of nitrogen in the form of nitrate is less than 100 milligram per liter. Then a biological treatment is good enough. If the levels are between 100 to 5000 milligram of nitrogen per liter, as in the sludge digestion liquor, again there is a biological treatment which is preferred. Okay, although ammonia stripping and the production of this are identified as interesting alternatives for resource recovery, that means to say that you just add a particular magnesium salt which can precipitate down this which can precipitate all the ammonia in the form of this magnesium ammonium phosphate salt. Okay, so pay special attention to it. By ammonia stripping, what we mean is that excess of nitrogen, it is measured in terms of the nitrogen as nitrate, but it could also be, nitrogen could be in the form of ammonium ions also. Okay, and if that is the case, then it can be stripped. Ammonia can be stripped by addition of a magnesium salt, eventually leading to production of this compound. However, if the level is more than 5000 milligram of nitrogen in the nitrate form per liter, then physicochemical methods are technically and economically feasible. Under these conditions, the biological treatment is not desirable. That is, don't think that you have a consortium of microorganisms which you will use for treatment of this level of nitrogen. Their physicochemical processes are required. A successful example is the steam stripping of wastewater with an ammonium concentration of 1.5%, followed by ammonia recovery, okay, which has been in operation on an industrial scale. So is this clear to everybody?
Yes, ma'am.
Yes, ma'am. Okay, so I think let's stop.
Like nitrogen, this nitrogen, we are talking about nitrogen in waste, right?
Nitrogen, which is coming in the wastewater effluent now.
Okay, ma'am. Yes.
Because it still contains a lot of nitrogen, because we will see the biochemistry of the activated sludge is such that it is a completely aerobic process. Okay, whereas conversion of nitrate into free nitrogen gas, it must occur in the absence of oxygen. And that is why that denitrification cannot occur in the activated sludge plant. All right, so we will come back, and in the next lecture I'm going to describe that what methods are available for nitrogen removal, and what kind of additions can be made to this standard wastewater treatment plant where the nitrogen can be recovered or how phosphate can be recovered. So that way these essential nutrients are recovered back from the waste. Okay, all right. So we will come back, and then in the next lecture, hopefully we are going to finish. And also by tomorrow, I'll give you out the results of the quiz. I'm done kind of halfway, and then I will just circulate it to everybody. So please go through. I will provide the reading material.